Hello students, today we are continuing with Biology class 12th, chapter number 4, Molecular Basis of Inheritance, revision lecture number 2. We have started with revision lectures in the previous class. Okay, so in our last lecture we have covered the topics, that is discovery of DNA and the genetic material DNA or protein. Okay, we have revised the topic about the discovery of DNA and the genetic material DNA or protein, where it was proved that genetic material is DNA and not the proteins, on the basis of 3 experiments that were carried out. One by one, we are going to revise all the 3 experiments. So, for proving that DNA as a genetic material, there were 3 experiments that were done. The first experiment was done by Frederick Griffith. Okay, so now we are starting with Griffith's experiment. First experiment, that is Griffith experiment. In the year 1928, Frederick Griffith studied the bacterium that causes pneumonia. The name of the bacteria was Streptococcus pneumonia or Pneumococcus bacteria. Okay, so remember this name of the bacteria for your exam purpose, objectives. Okay, the bacteria on which the Frederick Griffith have studied was Streptococcus pneumonia or Pneumococcus bacteria. Okay, this bacteria was responsible for causing pneumonia. What he has done, this experiment was done in the year 1928. Okay, he worked on the bacteria Streptococcus pneumonia. This bacteria Streptococcus pneumonia are having 2 types, that is R type and S type. We can say that it is having 2 strains, R strain and S strain. R type means rough type and S type means smooth type. A rough strain of the bacteria is non-pathogenic, non-virulent, whereas S type of bacteria is pathogenic and virulent. What do you mean by non-pathogenic, non-virulent? Non-pathogenic, non-virulent, rough bacteria means this bacteria are less harmful. They will not cause any effect on the host body where they are infected. But if any person is infected with S type of bacteria, then it becomes very dangerous for that person. This S type of bacteria is very pathogenic and virulent. It kills the person in which it enters. Okay? So now let us study stepwise steps of Frederick Griffith experiment. There were 4 steps in which the experiment was done. Okay, you can see here this diagram where the 4 steps are given. First step, R strain is injected into the mouse. Second step, smooth S strain is injected into the mouse. Third step, heat killed S strain is injected into the mouse. And the last step is rough strain, R strain, and heat killed S strain is injected into the mouse. You can see your result. What is happening? When R strain is injected into the mouse, what happens? The mouse is alive. The mouse lives. When S strain is injected into the mouse dies. You can see here the mouse has died. Then heat killed S strain was injected into the mouse, and the result was that the mouse lives. Mouse is alive. The reason behind this, we can say that heat killed S strain means what? The heating effect is given to the S type of bacteria, and because of the heating effect, the virulence have got over, and because of that this bacteria became safe. And in the last step, R strain plus heat killed S strain was injected into the mouse, and the resultant was that the mouse died. Then it was like question mark, how this mouse have died? So there was the further research that was done in this experiment. Okay, I repeat the steps. You can see here R strain is injected into the mice. The mice survived. That means R strain is non-pathogenic. Okay, are S strain injected into the mice died. It can be concluded that S strain is pathogenic and virulent, harmful bacteria. When heat-killed S type was injected into the mice survived again. It concluded that because of the heating effect, the bacteria became safe. And in the last step, heat-killed S-type plus R-type was injected into the mice died. When the blood samples of this dead mice was taken, it was observed that in large number living S-type of bacteria was obtained in the blood samples. What would be the reason? Okay, what had happened in the blood samples of the dead mice, large number of living S-type of bacteria was obtained. And from that, it was concluded that Griffith concluded that living R type of bacteria must have picked something from the surrounding heat-killed S type and got changed to S type. Okay, Griffith concluded that living R type of bacteria must have picked something from the surrounding heat-killed S type and got changed to S type. He named that something as transforming principle. This something was named as transforming principle. In the board exams, there can be a question that what is transforming principle, or it can be a question, explain transforming principle. So for this question, you have to write this Frederick Griffith experiment as your answer. The 4 steps that were carried out, the 4 steps you have to write in your paper. And then you have to write the conclusion that Griffith has given, that living R type has picked something from the surrounding heat-killed S type and got converted to S type. And then that something is named as transforming principle. Okay. Now let us study experiment number 2, that is Avery, MacLeod and McCarthy experiment. This experiment was done in the year 1944. That means it was done later, after 16 years later, after the Frederick Griffith. He has done in 1928, and then it was carried forward by Avery, MacLeod, McCarthy in 1944. What was done here? Avery, MacLeod and McCarthy has done, they have took the last sample, that is heat-killed S type plus R type, into a test tube. This time experiment was done in test tube assay. Okay, heat-killed S type was mixed with R type and then it was mixed with RNA. But what was found? There was no change in the test tube. Then heat-killed S type plus R type was mixed with proteins. Again, there was no change that was found in the test tube. Third time, heat-killed S type plus R type was mixed with DNA, and this time there was a change that was observed in the test tube. What was the change? The R type of bacteria got converted to S type of bacteria. Okay, R type of bacteria got converted to S type of bacteria. And because of that, what happened, that even after that, after conversion, protein digesting enzyme, that is protease, RNA digesting enzyme, that is RNase, was added, but there was no transformation that was observed. But when DNA digesting enzyme, DNase, was added, it has inhibited the transformation process. And hence the conclusion was given that this experiment proved that the transforming principle is nothing but it is DNA, that something that was used, that because of which the transformation has took place, that something was given the name as DNA. Okay, DNA, that is deoxyribose nucleic acid. At the conclusion was that the transforming principle is DNA. Okay, so for final conclusion, this experiments prove that the transforming principle is DNA. Okay, so in the first experiment we started to prove that genetic material, what is the genetic material, what is that is transformed, and in the second experiment we concluded that DNA is the transforming principle. Okay, so now one more experiment is there, that is Hershey and Chase experiment, and that experiment also is going to prove that DNA is the genetic material. Okay, but it has took the different things. Here we have used bacteria, that Hershey and Chase they have used virus, that we will revise in our next lecture. Okay, thank you for today. We will continue with our revision session in our next lectures.